I'm the PI for Advanced Rhode Island CTR, and it's my great pleasure today to introduce our speaker at our monthly clinical and translational research seminar series. This series is a monthly series, and we alternate between distinguished local scientists here in Rhode Island. We alternate their talks with talks given by our junior faculty research awardees. So today we're very honored to have Wafik El-Deiry speaking to us about translational therapeutics targeting cancer cell death. Wafik is Associate Dean for Oncologic Sciences at the Warren Alpert Medical School. He's Director of the Legorreta Cancer Center of Brown University and Menkoff Family University Professor of Medical Science at Brown. He's a practicing physician scientist and medical oncologist at Brown University Health, focusing on patients with colorectal cancer. And very importantly, he has done seminal translational research on cancer cell death, drug resistance, and in the process has developed new cancer drugs. So he really epitomizes translational research in this series. So again, we're very pleased and honored to have Wafik with us here today, and I'll turn things over to you, Wafik, and thank you very much.
All right. Thank you very much, Sharon, for the opportunity to present, and thank you for the kind introduction. This is a fun talk for me because I get to relive some memories and some lessons learned as I share them with you. But it's always fun to also speak about our research because I like to take every opportunity to get some feedback. Learning how to move the slides forward here. Okay, here we go. So these are my disclosures. So I'll give you a minute to look at them and range of different activities. So what I hope to do today is tell you a little bit about my career path, walk you through some of the key discoveries made throughout my career, tell you a little bit about entrepreneurial activities, our translational therapeutics, recent activity in the field of precision oncology and mitigating toxicities. I'll talk a little bit about networking and social media, and I'll end with some of the accomplishments and careers of individuals who have worked in my lab at Brown University since I came here in 2019. So this is the kind of slide I might make if I was trying to share something succinct, maybe trying to get a certain job, and you know, what are some of the experiences I may want to share with you. As Sharon mentioned, I am a practicing physician scientist oncologist. I am an American Cancer Society professor. There are about 50 or so ACS professors in the United States. I love translation and I love clinical medicine and oncology. I went into oncology because it's you get to take care of the whole patient, and I've always enjoyed that. And of course, there are many, many opportunities for basic and translational discoveries in oncology. So I went to medical school and graduate school at University of Miami. I did my oncology and medicine training at Hopkins. I was a Howard Hughes investigator early in my career. And then I went through a few leadership roles before I arrived at Brown University. For those who don't know about the cell cycle or the history of discovery, sometimes I do like to mention that I discovered a gene that I named WAF1, and nobody cared what you called a gene back in the early 90s because nobody thought it would be important, and it turned out to be important. And it's a highly cited paper, and I'll tell you a little bit about it. Um, and, you know, for many years, I thought that would be the most important thing I would ever do, which was sort of depressing. But then uh, got into the innate immune system and discovered a small molecule that went to clinical trials, and it's been shrinking brain cancer in humans. So, you know, living through that has been very, very exciting for me as a physician scientist. I founded several companies, and some of my recent activities include my service as chair of the WIN Consortium in Precision Oncology, journal editor, and senior member of the National Academy of Inventors. I can't give a talk without saying something about the Legorreta Cancer Center because that is a very important activity for me here, and I'm proud of our accomplishments over the last several years. And I just want to share with you that there's a lot of cancer in Rhode Island. We are in the top 10 states for your risk of developing cancer, and it's complicated. So if you go around the state, different cancers have their highest rates in different counties. We do have a very diverse state with different races, ethnicities, and ancestries. And so part of what a cancer center does is try to understand the causes so that we can prevent the cancers and develop better treatments and ultimately reduce the burden of cancer in Rhode Island and beyond as we make these discoveries. And our cancer center brings people together to collaborate and explores different priorities that are relevant to our vision and our mission here. And we also have an important educational mission. We have strengths in clinical trials, including first-in-human trials and community outreach activities within Rhode Island. So my story begins with my early childhood. So in the 60s and 70s. So those are my parents up at the top left, my late parents, and they were both physicians, and they came to the United States in the early 70s and brought me along and my brother. And so I grew up in that sort of immigrant environment in New York City with two parents who were physicians. You know, in the 60s, I enjoyed being at the beach and having fun, and always sort of thought I'd become a physician because my extended family has many, many, many physicians. In the end, I turned out different from the rest, and I usually have to explain to people when we have family gatherings, you know, what I do and why I do it. And I lived through that for many years. And now they just sort of leave me alone because they do understand the importance of research. So I grew up in South Florida after we lived in New York for a few years. And so I finished high school in Coral Gables, Florida. And there was a gifted program down there that placed students in different labs at the university or the medical school. So I ended up in the lab of the late Dr. Awad, who's a hematologist oncologist and director of the MD PhD program at the University of Miami, and started learning about protein biochemistry. And so that image there is human intestines. So he used to go to the operating room and bring back fresh human intestines, and we were isolating proteins. And so I got to spend time in the cold room learning about different kinds of chromatography and enzymatic assays, and sort of grew out from there as far as biochemistry, and was able to get on a couple of papers early in my career. And it's interesting, one of these papers is about protein acetylation and guanidination, and these are post-translational modifications that we were studying chemically at the time. And of course, acetylation now has a much broader relevance to gene expression and so on. I was a chemistry major in college, so I could have become a professional chemist, but it was really the influence of Dr. Awad that and my parents that sent me down the career path of a MD PhD student and physician scientist. When I got to the MD PhD program in Miami, I decided to work in the lab of the late Antero So, who was also a physician scientist. He was actually a Howard Hughes investigator in the 1970s, and his lab discovered DNA polymerase delta and also biochemically purified pCNA. And I got to work on E. coli DNA polymerase and mechanisms of mutagenesis. And this was a good experience because understanding DNA replication and mutations is really a good background for a career in cancer. When I was a medical resident at Johns Hopkins, I had the privilege of working in the lab of Steve Balin, who's a leader in the field of epigenetics. And I managed to convince the residency program director and the chair of medicine at the time to let me take a 3-month elective as a senior resident. And so I worked on and off for during my 3rd year of internal medicine residency in Steve Balin's lab. I got to set up the first PCR machine in his lab and used it to clone the human DNA methyltransferase gene and probed progression stages of colon cancer and looked at that worked. And what I learned from that experience had to do with how team science works. And there was much of that going on in that environment at the time with the Vogelstein lab, but, with biologists working with oncologists, working with pathologists and other basic scientists. And so those were really formative experiences. And I could have continued my career as an epigenetics researcher, but I really wanted to go and learn from Dr. Vogelstein. So when I was a medical resident, I subscribed to one journal. It was the New England Journal of Medicine. And, one day I got this issue, the September 1st, 1988 issue, and it had this paper, Genetic Alterations During Colorectal Tumor Development. And, it was by Dr. Vogelstein as the first author. And I read that paper, and it was elegant and was setting up the basis for the tumor progression model that has come to be known as the Vogelgram. And when I saw that, I said, this guy has the right idea. I need to go and learn from him. Um, he didn't make it easy to get into his lab. I already had a PhD. I wasn't going to cost him anything. But eventually, he did allow me to train with him. And in retrospect, this was one of the most important career moves for me and experiences. And he's been an amazing lifelong mentor and supporter of me, and I hope he gets the Nobel Prize at some point. So when I got to Bert's lab, we knew at the starting point at the time that the major tumor suppressor, P53, was just being recognized as a DNA binding protein. And I should say that this was in the early 90s. So P53 was discovered in 1979, both as a tumor antigen and as an SV40 T antigen interacting protein. And the whole world thought it was an oncogene for 10 years. I mean, Cell Science, Nature papers, because everybody was studying a mutated version of P53. And when I was starting my work in the Vogelstein lab, it had just been found that P53 could bind to DNA both specifically and non-specifically. And so Bert said, I would like you to figure out what P53 recognizes in the human genome so that if I show you a sequence of DNA, you will be able to tell me, will P53 bind to this or not? And so I set out on this project, doing something like we understand today as chromatin immunoprecipitation. And, it's cycled with PCR so we could amplify the genomic DNA fragments with linkers on the end so you could enrich for what was being pulled down by P53. And then I carried out footprinting, which is not really something people do today. We do footprinting a little bit differently with, ChIP-seq, but what you see there in the middle are assays, DNA methylation interference assays, where you could see protection of certain regions of DNA and basically read off the exact sequences of where P53 was binding. And so I did that for a whole bunch of genomic DNA pieces and had no idea what P53 could bind to, even after fine mapping, because the DNA binding site is somewhat promiscuous. And so it wasn't clear, but I spent weeks and weeks and weeks because of my background in DNA replication and the structure of DNA, was able to recognize common themes among the bases that make up this 20 base pair recognition element. And so we published this paper in the first issue of Nature Genetics, which, by the way, was a consolation because Nature didn't accept our paper, but it was in the first issue on page 45, and we never got preprints. We never got reprints of the paper. I actually had to buy the PDF a few years ago so I can have it and so I can make this slide that you're looking at. But the discovery of the binding site eventually led Nikola Pavlitic at Memorial Sloan Kettering to crystallize the DNA binding domain of P53 with its DNA response element. And the amazing thing, if you look on the right there, those residues shown in yellow that touch the DNA were residues that are mutated in cancer in P53. And, to this day, this remains as one of the strongest pieces of evidence that DNA binding and then other things that happen after that are relevant and important as to why and how p53 functions as a tumor suppressor gene. Now, I should say that mutations in p53 can be inherited in the Li-Fraumeni syndrome, where patients have developed multiple different common tumors in early adulthood. And we still don't, to this day, have a way of preventing those cancers. So basically, those patients end up getting a lot of screening. So, after the binding site, the plan was, let's do a chromosomal walk to find out what's near those binding sites that you've just isolated. And I really wasn't very excited because I knew I only had a subset of binding sites and I would be stuck with whatever I would find. And so I dragged my feet, and eventually Bert suggested I do a subtractive hybridization and a differential cDNA library screen, which I was open to and decided to do that. And fortunately, this led to the discovery of the P21 gene. And it was, there was a little bit of luck here because as you know, and some tumor suppressor genes or important genes that we end up studying are very, very large. But this cDNA was 2.1 kilobases, and it was a small protein. And so it kept coming up and being cloned over and over and over in the subtractive hybridization. It wasn't hard to get a full-length cDNA. And at the time we did this, we basically discovered it as a direct p53 target gene, and we had no idea how it functioned. It was really the work of Steve Elledge, who was then at Baylor College of Medicine, and Wade Harper. They're both now at Harvard, but at the time they had done a yeast to hybrid screen with CDK2 and discovered P21 as a CDK2 interacting protein, CYP1. And so it became clear it was one and the same. And then there was some work at Cold Spring Harbor that had found P21 in complexes of cyclins and CDKs. And it was beginning to be recognized as a regulatory subunit. Before these discoveries, our understanding of the cell cycle was just how it goes forward. We really had no understanding of the breaks on the cell cycle under cell stress or DNA damage. So that was clearly a good way to discover that mechanism. And so when I started my own lab, as a medical oncologist, I wanted to discover genes that were involved in cell death, because as an oncologist, I want to kill cancer cells. And so we used a subtractive hybridization and basically discovered death receptor 5 as a gene that is regulated directly by p53. And this gene was discovered at the same time as the cell death field was discovering DR5 as a receptor for the ligand TRAIL. TRAIL itself has gone through some clinical development. It's not an ideal drug because it has a very short half-life, but death receptor 5 agonist antibodies continue to be under development. So, we spent a number of years studying signaling in this pathway and what happens in cancer. And clearly, this pathway is not functioning as well as it should as part of the innate immune system in cancer. And, is death receptor 5 a tumor suppressor gene, and so on. And so I should say that before I left the Vogelstein lab, there were opportunities for collaboration, and I'm particularly proud of this one that is really not well known. But the reason I'm a co-author of this paper is because I suggested looking at DNA polymerase mutations in cancer as another mechanism of high mutation frequency, as in mismatch repair defects. And I arranged a collaboration between my thesis lab and my postdoc lab, where DNA polymerase delta variants were found in some tumors with the replication error phenotype. And of course, subsequently, other polymerases have been found to be mutated in tumors with high tumor mutation burden. Okay, I think, let's see. Having trouble here working these slides. Okay, so those were the early years, and then I became a faculty member. I remained at Penn for a number of years. You know, the real fun of publication and impact happened in subsequent years. But I will point out a couple of things on this slide that I made up a few years ago here at Brown. You see right there this plateau. This can happen in your career where you're at a place and you're in a plateau. You're surviving, but maybe your career isn't exactly where you'd like it to be. And, you know, I stayed at Penn for 16 years, and then I moved a couple of times before I came to Brown. And I attribute, you know, these increases in citations and impact as a result of that sort of renewed energy and resources that are important in our career to help us achieve our goals. So, you know, my lab goes back to 1994. It's been at a number of institutions. Because I'm interested in cancer therapeutics, we take a holistic approach to cancer. We always think about translation and moving discoveries to clinical trials. It is technology enabled. We have a very diverse group. We collaborate internally and externally. And we really want to have impact in the area of translational therapeutics. So, you know, cancer is complex. And has many hallmarks. And on the left are the four newest hallmarks of epigenetic reprogramming, phenotypic plasticity, the microbiome, and senescence. And there's a lot of heterogeneity. There's clinical heterogeneity, there's tumor heterogeneity, there's host heterogeneity. Ultimately, this is a great opportunity, but it's also a sign that in the end, you know, precision medicine is an important path for our field. And I will come back to that theme a little bit later. So the path from discovery to translation took decades, basically, with the early work, with the discovery of death receptor 5, and then later with drug screening to find molecules that upregulate this pathway, to the discovery of the trail-inducing compound that we made in 2007, to the moving of that small molecule to clinical trials by 2014. And it was by 2017 that it was being recognized to have activity against brain tumors with H3K27 M mutations. I will say that in a way, this is a precision medicine story because the initial clinical trials were done in solid tumors as well as brain tumors. So the first brain tumor study was done in Boston and enrolled 17 patients, and 2 of those patients had exceptional responses. And this is how it was recognized that brain tumors with H3K27M mutations, these middle of the brain diffuse gliomas that are very aggressive, not operable, don't respond to anything really, and have short survival, were actually responding. Now, so in the way precision medicine works efficiently, one can then design and conduct a clinical trial that enriches for patients with tumors that have the specific mutation against which you have a drug that is active. And you could see up here this paper that came out in 2024 about the data in a population of patients with glioma and H3K27M being treated with ONC201. The original drug we call TIC10 for trail-inducing compound number 10. And it's clear that the majority of patients either have a response or have disease control, but clearly a bunch of patients do not respond. And the resistance mechanisms have been under study and continue to be. Our approach here has been to ask a lot of questions, because when we discovered this small molecule, we didn't specifically discover it as a small molecule to treat brain cancer. It turned out to have this activity, and been asking the question ever since, why did it turn out that way? And how can we get it to work better in brain cancer? And how can we get it to work better in other tumor types? So as I said, the preclinical data showed activity across tumor types, brain cancer, colorectal cancer, lung cancer, lymphoma, breast cancer. And but that really didn't predict what happened in the clinic. We didn't have enough money, and I founded a company called Oncoceutics that I mentioned that licensed the compound. The company didn't have enough money to at the time, do all of the required studies the FDA needed to allow optimal dosing early on. So as it turned out, in the first in human studies, this drug was given to patients every 3 weeks. Imagine you have a brain tumor in the brain tumor trial, and you have this aggressive growing brain tumor, and you're being asked to enroll in a clinical trial, and you get to take this pill every 3 weeks. It's a little strange, but the results showed activity even at that dosing. Later on, we did dose intensification, which brought out immune effects and brought out some effects on suppressing metastasis. I should say that the first in human study had signal in prostate cancer as well as endometrial cancer, and there is work out there that has shown activity of ONC201 against neuroendocrine tumors. There's never been a phase 2 study in prostate cancer, and so I'll touch on that. The drug is on track for approval, hopefully by August of this year. It really could have been approved several years ago, but the approval requires a lot more than just the efficacy in clinical trials. So it has taken longer, but it's on track. Now, the mechanisms are novel and interesting, and the clearest mechanism of how ONC201 or this other analogs act is as ClpP agonists. So ClpP is a mitochondrial casinolytic protease, and an agonist of that protease activates it, And then you have proteolysis within the mitochondria that shuts down oxidative phosphorylation. And there's a signal that comes out of the mitochondria to a kinase known as HRI, and this acts on EIF2 alpha. So HRI is part of is one of the kinases that are part of the integrated stress response. Others include GCN2 and PKR and some others. But ultimately, what happens in this response is the global suppression of protein translation. And then there's also the activation of specific transcription of genes through ATF4 and CHOP, which are transcription factors in the integrated stress response that lead to activation of specific genes. And so it's taken us some time to recognize that the suppression of translation works favorably in some mechanisms. So there was a time when we asked the question, how is it that H3K27 that's mutated to a methionine, why is that sensitive? And could we mimic that by inhibiting the PRC complex that has EZH1, EZH2? Could this synergize and so on? And we discovered that there is synergy between the imipridones and EZH2 or HDAC inhibitors. But we recently reported that these imipridones, most likely through suppression of global translation, actually inhibit EZH1 and EZH2. But across tumor types, where we've looked at the combo, there is synergy. So the suppression is not enough. And when you come in with a EZH2 or a dual EZH1, EZH2 inhibitor, you get potent synergy. We noticed that the synergies and death-inducing effects were more associated with increased acetylation of H3K27 versus methylation. And this is an area we continue to work in because the exact nature of the methylation versus acetylation at this lysine and phenotype of these diffuse gliomas and the response to imipridones continues to evolve as we understand more. We recognized last summer that the imipridones suppress MGMT expression, which I think is relevant and significant for their use in brain cancer because MGMT is a known resistance mechanism to temozolomide, which is the mainstay of therapy in combination with radiation in glioblastoma, not the H3K27M variety. Interestingly, the H3K27M variety is known not to respond to temozolomide. So temozolomide is excluded, but I think that maybe overlooking the fact that the combination of the imipridones and temozolomide is associated with suppression of this resistance mechanism. So, we're working on prioritizing some future trials. This is like a 2-year experiment done by Lan Zhu, looking at the triple combination of either ONC201 or a more potent analog 206, temozolomide and radiotherapy. And the triple combo had the most surviving months well into 2/3 of a year here. And so this is something that we are trying to translate, and likely by the time it gets done in the clinic, it'll happen with ONC206 versus ONC201, but the paper just came out a few months ago. We are working on translation with some other combinations, particularly interested in prostate cancer because there is a signal with monotherapy with ONC201. There's never been a phase 2 trial. We've collaborated with Benny Carnero for a number of years on combinations with enzalutamide or darolutamide, and are working on a concept where this can be done in the clinic. I think these types of trials will become more feasible and possible to do, especially after the drug is approved. We have looked in brain and other tumors, as I said, about the epigenetic inhibitors in combination with imipridones. And we have been working on analogs. So 206 is already in clinical trials in brain cancer. 212, which is even more potent, has not entered the clinic, and we have been working on preclinical studies for early clinical testing with that analog. Now, I'm going to take like 2 minutes and tell you that there's other stuff that goes on in my lab, and some of it has led to entrepreneurial activity. And it would literally take me hours to talk about some of these different areas. But the P53 theme is there, and one of the directions I'm very interested in is in restoring a P53 pathway in tumors with P53 mutations. And interestingly, we have small molecules that can do this. Some act through P73, which is related to P53. Others, interestingly, activate the integrated stress response, but not through CLPP that I told you about, but are tapping into that same type of mechanism, which I think is very interesting. We've also had, I mentioned earlier, we take a global approach to cancer. So hypoxia is a major mechanism of resistance to therapeutics, and it's a breeding ground for selection of more aggressive tumor cells and therapy-resistant cells. And so for a number of years, we tried to target hypoxic cells. We've carried out, we and others, and hypoxic, and we uncovered small molecules that could sensitize hypoxic cells to killing in combination with other drugs. And eventually, in the process of trying to translate, we demonstrated that CDK4 and 6 inhibitors that are used to treat breast cancer can destabilize HIF-1 alpha. And then former graduate student Shui Zhao did a proteomic screen and discovered an E3 ligase called SMRF2 that associates specifically with HIF-1 alpha in CDK4/6 inhibitor treated cells. And so this has sort of opened up an area of interest in the lab of regulation of HIF-1 alpha by kinases, such as cyclin-dependent kinases or MAP kinases, and that these are non-canonical VHL independent ways of regulating HIF-1 alpha. So, we have ways of manipulating HIF-1 alpha for therapeutic gain, and that's what SMRF is focused on. Happy to talk more. This is a concept related to resuscitating drugs and bringing them back to the clinic, another entrepreneurial activity. Some of the lessons I've learned is it doesn't get easier to raise money for entrepreneurial activity. Investors will always be skeptical, and they will miss great opportunities. Some things do get easier. We network, we know people, and we get better at building teams and protecting IP. And of course, de-risking is on everyone's mind if you want to translate and move things into industry. Persistence and resilience go far. I will add another one that my chair of pathology has instilled in me, which is persuasiveness, the power of persuasion. So, persistence, resilience, and persuasiveness are really useful in this type of career. And perhaps the last is really important as far as getting grants and convincing people to invest in your work. So I'm going to spend a few minutes talking about some really cool new data that links the innate immune system to DNA damage, induced inflammation and fibrosis in vivo. So a number of years ago, we created a death receptor 5 knockout mouse and showed that it was radio resistant. And we gave a cohort of these mice a sublethal dose of gamma radiation and showed that they started dying off, between 4 and 8 months. And we uncovered inflammation and fibrosis in the lungs as well as other organs. So we've known since we published this paper in 2008 that the trail pathway suppresses this inflammation and fibrosis. And of course, if you look at the literature, it's all about TGF beta. And, for years, I didn't feel like I could ask someone in my lab to administer trail, which has a very short half-life, 2 or 3 times a week for 6 or 8 months. It just seemed like it would be too much. Eventually, I came to Brown and convinced a company to fund some studies for a new trail formulation called TLY-012, which is pegylated to give it a longer half-life, and work started by Anna Louie, a surgery resident, and continued by Jill Strandberg, who actually did the vast majority of the experiments in this JCI paper that was just published in 2025. And there was a surprising result for me, and I'm sort of kicking myself. I wish I had done this experiment years ago. It turned out that the rescue from the radiation-induced inflammation and fibrosis was just a lot easier than we thought it would. So in any event, we designed this experiment, and this time, our original JCI paper had just DR5 null and wild type mice. This time we added trail null mice, and we predicted that ONC201, which is a trail-inducing compound, or this TLY-012 preparation would rescue wild type mice. They would not rescue DR5 deficient mice, and that only the trail formulation would rescue the trail null mice. And this is what happened. I mean, the phenotypes were worse in the DR5 null and in the trail null, not shown here. We'll show on the next slide. But there was rescue. This was the very first experiment that was done by Anna Louie before she left Brown to complete her surgery residency at Duke. But Jill did these experiments in both male and female mice, trail deficient and DR5 deficient wild type mice. And basically the predictions are all recapitulated here. I should say that the phenotype is much more severe in the female mice, and that's an emerging direction for the lab. Not only was the acute inflammation rescued, but the lethality of the mice was rescued. For female mice, we had to use lower doses of radiation, otherwise it was just too toxic than the male mice. And very high doses of radiation are just not possible to rescue. We observed for the first time radiation-induced dermatitis that came out in the trail null mice, and it was also rescued by TLY-012. And we also observed rescue of a radiation-induced esophagitis, which is a big problem for patients with esophageal cancer who get radiation, and they have all kinds of issues with the severe esophagitis. So these experiments are promising and included use of ONC212, which is a trail pathway agonist. We have some insights into the sex differences in severity of pneumonitis with this X-linked TLR7 gene. TLR7 is linked to interferon gamma and macrophage responses, and this is an area of ongoing interest and investigation. I should say that the TLY-012 rescued fibrosis as well, chronic fibrosis, 22 weeks. The trichrome stain is the blue on the left, showing the fibrosis, and the rescue on the right, showing much less blue. There was improvement in lung area by mouse CT imaging and cross-sectional areas, as you can see here. We did do a tumor-bearing orthotopic syngeneic model with breast cancer in the mammary fat pad, and showed that irradiation alone was effective. The addition of either TLY or ONC or the combo did not mitigate the activity. Of course, we fully predicted that, but that mice were rescued from respiratory decline with these innate immune pathway agonists. We noticed that CCL22 is reduced in association with rescue by TLY-012. We also noticed that we could get some amount of rescue even post-irradiation exposure, and this is of interest in the world of radiation countermeasures. Jill conducted a preliminary study targeting CCL22 to see if it could cause rescue of mice with severe pneumonitis. And I'm remembering correctly, these are trail null mice. Yes, it says so right there. So this may be an independent parallel pathway, but there are many, many questions. So, some of our directions include understanding better the links between the trail innate immune pathway, TGF-beta, CCL22. We saw other cytokine changes we'd like to follow up on. Obviously, combinatorial strategies to treat the pneumonitis are of interest, as are other causes of pneumonitis, including bleomycin, which is also used in a not so good mouse model of idiopathic pulmonary fibrosis. But we're also interested in immune checkpoint blockade-induced pneumonitis, which is a significant problem in oncology, especially when you start combining immune checkpoint blockers. I think the esophagitis and dermatitis and sex differences do have a future. So I've got just a bunch of slides that are going to go by really, really quickly. And it's about networking and about social media. I have a strong presence on social media, and I appreciate it very much because I learn a lot from it, and it's a great way to meet people and network with people. And so, each of us should be asking ourselves, these questions. What is our network? How big is it? And how can we grow it? And, what areas do we network in that would be helpful to us professionally? I think of social media as the world's largest neural network. It's just simply amazing, and it's very efficient at information exchange for things we're interested in, and it's really great for learning. So it's part of the reason I use it so much. So, it's a great way to learn the latest in our field and follow leaders in the field and connect and build online communities, find collaborators across academia, industry, government, find mentors, build your reputation, share knowledge. Always got to respect others and be careful with private information, political agendas, offending others, and don't get caught up in senseless arguments. I network to learn and impact in precision oncology. I mentioned the WIN Consortium in personalized cancer medicine. I think this is the future of oncology, and it's going to be more mainstream. It may take 20 years. Hopefully, it'll take less time. Efforts have been in the field for about 15 years now, and there's evidence that N of 1 personalized therapy combinations do help patients survive longer with each patient as their own control. And I'm not going to go through this, but I'm happy to elaborate if there are any questions. I think I mentioned this. So I'm just going to show these slides of some trainees. This guy, Akash, was like waiting outside the door when I arrived at Brown. He wanted to get his master's in my lab and eventually ended up going to med school. Along the way, he won an award for academic accomplishment. His research was in the area of pancreatic cancer and therapy combinations targeting the trail pathway. Francesca was interested in senescence and senolytic therapies. She ended up going to graduate school at the Rockefeller, and this is her presenting at AACR. William McDonald is currently a student at Columbia, a medical student, and his research was in integrin signaling as a therapeutic target. Praveen is currently a medical resident at Cornell. You couldn't get Praveen to stop talking about ferroptosis, okay? He just is so interested in that pathway and has made very nice contributions. I tried to convince him to go for a combined degree, but he's determined to pursue a certain career path. And I'd look out for this guy because he's going to do great things. Kelsey and Lindsay got Scholar and Training Awards at the AACR in 2022. Lindsay also won an International Poster Award in Barcelona, and Kelsey was awarded an NCI F31 after her first submission from Brown University. Thomas Brown, an undergrad, ended up in med school at Tufts. Max ended up in a surgery residency, and Vita is about to start a medicine residency. Shui, who worked in hypoxia, is currently a postdoc at Dana-Farber. Josephine went to med school at the Cleveland Clinic. Ilias is currently a faculty at MGH, and very recently, Brooke Verschleiser just won the Llewellyn B. Clapp Prize for Outstanding Undergraduate Thesis from the Chemistry Department. I think a lot about immune cells because they're so important. They're really our last hope. Getting rid of the immune suppressive tumor microenvironment and boosting the tumor suppressive pathways. And you can order a t-shirt just like that. Our students get out there and meet people. So one of our summer students sent me this 3 years ago. I'm sure I don't have to tell you who the guy on the right is, because he now summarizes papers in the New England Journal very nicely. Um, and we work at the Ship Street Labs, and there's a band that comes and plays. They're known as the Extraordinary Rendition Band, and they're usually out here playing, and it's a lot of fun to listen to them. And this reminds me that we need a work-life balance. I became a boat captain during COVID and enjoy uh, being on the water. And this is our research group with work that I highlighted, mentioning people's names as I talked about them. So thank you very much for attending, and I'm happy to answer questions.
Fantastic. Thank you so much. That was really a wonderful talk and inspirational, in fact. So we have a lot of questions. How do you, Sean Lawler asks a interesting one. How do you see academic and translational science in the US changing or adapting in the next few years?
I think we're all going to have to adapt. Some of us have been adapting for years and diversifying our sources of funding. I think we need to help patients ultimately, and we need to, academia is a pipeline for discovery. And I think some things are going to need to change in terms of industry playing more of a role in supporting research and translation, because it's not an even playing field. They take intellectual property that the government invested in and essentially develop it to help their shareholders and sell drugs at very high prices. So those are really some big problems. I do think some of the evolution is going to have to involve finding other sources of funding to facilitate the translation. I have certain views. I could talk for a while about pharma influence on clinical trials and so on, and also like what clinical trials the government should support. I actually think the government should support innovative clinical trials and not um, derivative trials. But I don't know if that answers your question, Sean, but that's what I'm thinking.
And Nicholas Sarlis asks, would TLY-012 also be of use to increase the therapeutic index of radioligand therapy, considering the increasing popularity of theranostics?
So I'll tell you, TLY has great potential as a cancer therapeutic in combination with a variety of other agents. We have combined it with immune checkpoint blockade. We have combined it with ONC201. ONC201 is a trail-inducing compound. And when we first discovered it, you would ask me this question. I say, well, the small molecule upregulates trail. Why do we need a trail agonist? Or why do we need a trail formulation? And what we learned over the years is that the cells treated with the small molecule don't make enough trail. And when you combine the small molecule with trail, it's like flipping a switch where DR5 goes up, and now the cells are primed for killing by trail. I mean, trail, Nick, has been studied for 25 years now, and there is synergy with many pathways. As far as radioligands, sure. I mean, there's synergy with other other cancer suppressive therapeutics. We demonstrated some synergy between the small molecule and radiotherapy in that GBM study, but yeah, I think there's every reason to. The issue with TLY-012 is being developed by D&D Pharma initially for pancreatitis and systemic sclerosis as fibrosing illnesses, but it hasn't yet entered the clinic because for business reasons, companies sometimes prioritize other therapeutics. And for this reason, we would hope that it would go into the clinic, but I've also been interested in trail pathway agonist antibodies, DR5 agonists. Many companies have them. And I'm currently collaborating with a startup. We just submitted an SBIR on a way to make antibodies in corn for oral administration, if you can believe that. But so I think there are ways to, this pathway, which I really believe in, has not been exploited enough in cancer therapy.
So I'll ask a question. You discovered some serendipitous extrapulmonary positive effects of the drugs with the esophagitis and dermatitis, but that also points out that the drugs are acting systemically. And in order to really screen for systemic effects, that's the kind of grunt work that industry is really good at. How does an investigator go about looking for unanticipated systemic side effects of agents like this?
Well, I'll just start with, I'm sure you're well aware of things coming out of the FDA about alternative models to animals. I personally think that understanding the systemic effects of novel therapeutics is hugely important. And it's, even if you go up to monkeys, it's not always predictable what will happen in humans. And this is why, there were some incidents years ago where 6 patients get a drug and bad things happen to all of them. And so we don't do that anymore. You start with 1. Okay, but I would not want to be the one who consents for a novel agent that has never been tested in animals in a first-in-human study. I just think, the ethics of it have been worked out for many decades. And I think, if the organoids or organ on a chip models or all these things were really up to par in predicting systemic effects. I mean, you can predict, obviously, with cytokines that can have systemic effects, but I think it's just way too complex. And I don't think I'm skeptical, and I would, caution about getting away from this, even if these technologies complement each other, but I don't think we're ready to get away from the in vivo models.
Okay, great. Well, there's other questions. And Roxanne asks, in addition to waiting outside your door, how does one become a member of the El-Deiry lab? And then I'm afraid we're going to have to stop after that one.
People are always welcome to join us at lab meeting. And, Roxanne, I was thinking of reaching out to you very soon because we have some really cool preclinical translational work in small cell lung cancer that I want to talk to you about. And Humira is part of this group, and Jerry Azzoli. And I think it'll be fun to look at where this is going. It may be a budding Bruag study.
Fantastic. Well, thank you so much. This was intriguing, fun, and As a pulmonologist, I'm looking forward to seeing the studies on bleomycin and perhaps TGF-beta induced models of idiopathic pulmonary fibrosis, because it seems like these drugs may have treatment options for non-malignant fibrotic diseases. So anyway.
And I did want to thank you, Dr. Rounds, as one of our co-authors on this nice JCI paper. Your input and rigor with regard to the field of pulmonology is truly what multidisciplinary collaboration is all about.
That's a lot of fun. Okay, thanks a lot.
Thank you,
everybody.